Welcome at the webinar Challenges and Developments in the Field of Therapeutic Radiopharmaceuticals. This webinar is organized by Working Group 7 of Eurados, and my name is Ramona Bouwman, and I'm employed at the Nuclear Research and Consultancy Group NRG in Arnhem, the Netherlands. I'm the leader of the task group Internal Dysmetry of Therapeutic Radiopharmaceuticals as well. Today's webinar consists out of 5 short lectures. After a short introduction on Eurados, I will start with the first lecture, which provides some background on the task we are working on. Subsequently, 4 invited experts involved in the production will proceed and explain something about the challenges and developments of therapeutic radiopharmaceuticals. If you have any question, you can ask them via the Q&A box. If there is time after a lecture, we'll try to answer them directly, and otherwise we will answer them in the Q&A box. So starting with a short introduction about Eurados. In brief, Eurados is a network organization consisting out of 80 voting members from 31 countries and is chaired by Philip van Haveren from SCK Sen in Belgium. The Eurados office is operated by Kirsten Heurkamp, Babs van Hees and Helmut Schumacher and Eurados has a council of 12 people taking care of the management of Eurados. Eurados is a network organization who are active in the field of dosimetry, and the work is organized within a variable number of Eurados working groups. Currently, 8 working groups are active, and in 2021, a new working group is started, pilot group is started called Dosimetry in Nuclear Medicine. Today's lecture will only focus on working group 7, internal dosimetry. The strategic research agenda of Eurados guides the working group, the work done by the different working groups, but is also used in the priority setting of research goals within the European Commission. The latest version of the SRA is from August 2020 and can be found on our website. So moving on to Working Group 7, Internal Dosimetry. This working group is led by Bastiaan Breustad. Within our working group, we are focusing on internal dose assessments and intercomparison exercise with case studies, where we have defined different focusing points like individual monitoring, biokinetics, dose assessment methodologies, emergency situations, micro and nano dosimetry, uncertainties, and training and knowledge management. These focus points form the basis for the work done by our task group. Example of the current task within our working group is the OER guidance report, which should include all information necessary for the implementation of the ICRP biokinetic and dosimetric models, which have been revised recently. Or the IFIC project, a European project to assess the implementation of the requirements for individual monitoring, or the hot particles task for the characterization of small high active radioactive particles, so-called hot particles. And of course, important for this webinar, internal dosimetry of therapeutic radiopharmaceuticals. So let's start with the first lecture. I already explained who I am, but I would also like to acknowledge that the work done within this working task group, I cannot do it myself. So here you find a list of all those who are helping me. During this lecture, I would like to give you some background on the task, but also provide you the goal on the task we are working on. Subsequently, I would like to explain in brief the organization of the review that we are currently working on and a brief look to the future on how we would like to proceed. We initiated this task after the publication of the new Eurados SRA in 2020, which has a dedicated vision on internal dosimetry of therapeutic radiopharmaceutical, namely Vision 4, towards integrated personalized dosimetry in medical applications. With the knowledge and experience of Working Group 7 in, for example, Biokinetic modeling, micro and nano dosimetry, but also on dose and dose effect relationships, we believe that we can contribute to this vision, and then especially for the development of knowledge on internal dosimetry of novel radiopharmaceuticals emitting alpha, beta, and Auger radiation, and also on the transition towards internal dosimetry in clinical molecular radiotherapy. But you can imagine that this is a comprehensive task involving a lot of different disciplines, and that the question we had is how and at which point can we contribute to the visions and what are others doing? To answer this question, we decided to start with a review to summarize current status and challenges that need to be addressed before a methodology for integrated personalized dosimetry can be proposed. So in summary, the goal of our task is to find the roadmap for dosimetry in radionuclide therapy based on individual physiological and biokinetic parameters, drug-specific parameters, and dose estimates up to local subcellular levels, so that we can provide a patient-specific estimate of the dose, and this should provide us guidance on how we as Eurados can proceed with Challenge 4.2 of the SRA. We started with a review, and there we focus on the three important points for Working Group 7, namely biokinetics, dosimetry, and dose effect relationships. Besides this review, we also search for collaboration with other umbrella or experts or network organizations which are active in this topic. We divided the review into 4 chapters. A general introduction to the challenges and the development within the field, a chapter about biokinetic modeling, about dosimetry, and a chapter about dose effects in therapy in general. For each chapter, 1 or 2 persons will take the lead to collect all relevant papers, and subsequently, with the other task group members, we read the papers and write the text of the report. Chapter 1 is led by Mario Medvedek, and Siriam Medici will start with looking into the history and development in dosimetry for radionuclide therapy, the challenges and developments for today's clinic, but also going into detail about the physiological and biological properties of radiopharmaceuticals and the development and challenges in the production of radiopharmaceuticals. But since we are from Eurados not a network organization on the production of radiopharmaceuticals, we decided to organize this webinar and invite several experts from the production sites, and we are happy that they took this opportunity to tell you something about the current development and challenges. Chapter 2 is led by Kirsten Hurkamp and myself, and starts with an introduction on biokinetic modeling and based on the ICRP and MRD models. Subsequently, we provide an overview of the current approaches for biokinetic data collection and support this with a summary of data for a single therapy. In this chapter, we would also like to go into detail about PBK modeling, physiological-based pharmacokinetic modeling. Similar to the models of ICRP and MRD, these models are compartmental models of the human body, but in contradiction, the transfer parameters are based on physiological and pharmacological principles rather than fitted transfer rates. PBK modeling is already known for several decades in the research and development within the pharmaceutical industry, but only since the last decade it explored in the field of the radionuclide therapy. With this type of models, we hope that in the future we can model the position of the radiopharmaceutical over time inside the human body up to cellular level where needed. Chapter 3, Dosimetry, is led by Vivo Lee, and together with the other task group members, they will provide an overview of the formalisms used in nuclear medicine, like for example the absorbed dose and the developments over time. Traditionally, dosimetry is conducted on whole body or organ level, but they will also go into a depth about dosimetry on subtissue level towards cellular level, so-called micro and nano dosimetry. Subsequently, they will also tell something about different patient-specific dosimetry methodologies that are currently being used, like the MIRT procedures, voxel-wise approaches, and 3D Monte Carlo methods. Chapter 3 will be finalized with a discussion on software and tools available for dosimetric evaluations. In Chapter 4, led by David Broggio and Balas Madas, dose-effects relationship in therapy will be discussed. Summaries started with a summary on dose response models in external radiotherapy, like the linear quadratic model and tumor and normal tissue response models. Subsequently, they discussed the differences between external radiotherapy and radionuclide therapy. It is discussed, and this discussion will be finalized with a debate on pros and cons about dose-driven radionuclide therapy. So like you can see, the review is still work in progress, and we hope that we can finish the EURODOS report by the end of this year. We envision that if we can model the distribution and the associated energy deposition up to subcell level, we can help to move forward evidence-based personalized treatment procedures for radionuclide therapy. After the report is finished, we would like to disseminate the findings and search for collaborations and/or consortia to proceed with the proposed challenges and developments. So I hope that you now have a feeling on the work that we are doing within our task group, and please feel free to contact me via the chat or later via mail or phone. So thank you for your attention, and feel free to ask any questions in the Q&A box. In the meantime, we also have a question for you, and we launched a poll so you can give us feedback from your opinion about the role of EURODOS in the development of internal dosimetry of therapeutic radiopharmaceuticals. So now quickly check whether there are questions in the Q&A box. For so far, I don't see any questions yet. So therefore, I would like to propose to go to the second lecture. The second lecture is given by Sabine Sitzman-Kolbe from Bayer, and she will inform us about thorium-227 labeled traces. Sabine, the floor is yours.
Yes, hi, and I want to thank the organizers of EURODOS for inviting me to give a presentation. My name is Sabine Zitzmann-Kolbe. I'm a biologist, so don't expect too much mathematics from me, and I hope it will still be interesting. And I was going to tell you about the research we're doing with thorium-227 and what we do with developing new tracers. So this is what I have to disclose. Of course, I'm working for Bayer AG in Berlin and everything else. So to get started, as a short introduction, Bayer has 3 areas where we work in the field of oncology. One is targeted alpha therapies, oncogenic signaling, and immuno-oncology. And the targeted alpha therapies actually started with radium-223, Xofigo, which is was the first and only launched alpha therapy approved, and it's now distributed in quite a lot of countries. And of course, it has its restrictions, as it can only be used by or for the treatment of bone metastases, as it kind of enriches in the bone because of its physical chemical properties. And there was the question, now can we use alpha therapy also for other indications? And the problem is the radium does not chelate. So there were a lot of ideas how to use alpha therapy also for systemic radiotherapy and also for soft tissue disease and metastases. And as you are aware, there are several um, specific properties and we think advantages of using alpha radiation. On the left, you can see a schematic showing like the main feature is that you deliver very dense damage to the cells, to the DNA, with a lot of double DNA strand breaks. That you have to use much lower doses, and you have a limited exposure to the surrounding tissue due to this very short penetration distance of the alphas. And for targeted alpha therapies, of course, that comes also from the field of using beta. And there are lots of literature and experience out there that you can, for example, use antibodies and small molecules to deliver the alpha to the tumor, to the metastases. And there are also quite some examples in the literature here. And what we have been focusing on was first have an antibody that targets specific, and then you have a linker and a chelator. And in this chelator, you chelate the radiotherapeutic. And here we have been focusing on thorium-227, as so far we have not been able to get the radium-223 into a chelator and make it stay there, at least not for as long as we would need it for a therapeutic time. And so on the left, you see the radium, the thorium-227, which is actually the mother of the radium-223, which we have been using for the targeted alpha therapies, or we call them TTC, so targeted thorium conjugates. And out of the thorium-227, then you have the radium-223, and then you have the long chain of daughters that is basically the same that comes out of the radium-223, of course. And the thorium-227 has a half-life of 18.7 days, and it can be stably complexed via Hopewell chelators. And we have a supply that comes also from the production of the radium-223. So that seemed quite good for us as a starting point to have a therapy there. On the right, on the left, you see also the actinium-225, which is, I think, the most used trace alpha at the moment used for therapy. In principle, the actinium-225 is quite more similar to the radium-223, so it has also a half-life of around 10 days, and it has only short-lived daughters. For the thorium, you have, of course, the 18.7 days, and then the first daughter is the radium-223 with a longer half-life before all the short daughters come. For the actinium, you only have the short daughters, with bismuth having the longest lived daughter with 45 minutes. So when you use actinium-225, you deliver a faster dose rate to the tumors, but you always have, at least at the moment, the secular equilibrium to work with. But now I would like to concentrate first on the thorium-227 and give you some insights on what we have been working on. So we have currently two projects that are in phase 1 dose escalation. One is a PSMA TTC, so a PSMA targeted thorium compound. That's an antibody from Progenics Pharmaceuticals, and with a Hopewell chelator, it has been shown very strong preclinical efficacy, and has also been in various models, and is currently in a phase 1 where we have dose escalation, and we can are at quite considerable doses at the moment. On the schematic on the right, you see the effects you have when you have a comparison. It's in black is the vehicle, and here we have a combination with darolutamide. So in green, you see actually the TTC by itself. And when you combine it with darolutamide, which is a compound heavily used also in prostate cancer patients, you see that you can increase the efficacy of the alpha therapy very strongly. We know for the one thing, this is due to an increase of receptor on the cells. So you have more target on the cell, which makes you deliver more radioactivity to the tumor, but also some other additive effects. The other project we have here is in the phase 0 study is a HER2 TTC, is a trastuzumab biosimilar. And this project, HER2 is not a new idea, but the idea with the TTC was to use the strong effect from the alpha therapies to also be able to use this on HER2 low expressing or even resistant tumors. And what you can see on the schematic on the lower right is when you have a model that only has an IHC score of 1, so it's really a low expressor, which you would not use for a conventional HER2 therapy. And when you see in black, you have the saline. And when you look at the blue curve, you see that's the HER2 TTC, which delivers quite some dose and elicits a response to the tumor. So we think that here with the alpha would offer an additional treatment for the patients. So both of these are currently in a phase 1 in clinics. And I was now going to focus on the PSMA TTC as an example, how we got there and what are the special requirements, and also the research we do to investigate the effects of alpha on the tumor cells and on the cells itself, and also when it comes to the resistant or the sensitivity of the cells and why that is somehow different. So first thing, of course, you need is to have a good target. And we're not going to discuss this today, but that's like the most crucial thing, that you need a very good and specific target. And then comes the next big challenge to find a good targeting molecule. And here in the lower left, you see that, of course, what you need is you need to have very good binding. So what you first test is, does your antibody bind? And does your antibody also bind when you have the chelator on the antibody, and even when you have radioisotope in the chelator? So you should not prevent the binding. And then right next to it, figure B is, we think it's very good if you also have internalization of your targeting molecule. Because what happens when you have your first decay, then it's very likely that your alpha leaves the chelator, and then you have an alpha where you have 4 more potential alphas that could contribute to the dose delivering to the tumor, and that might get lost while they diffuse away or go somewhere else, and that would not just reduce your dose, but it could also elicit more side effects, because that's another big thing that's interesting. So where do the other daughters go, the ones that are generated and that are probably also not in the chelator? And after, so these are like the basic requirements. And then we check for other effects these targeting molecule with the alpha elicits on the tumor cells. For example, we look what are, or do we induce double strand breaks on the cells? Is that dose dependent? And also when we check cell cycle analysis. So how do the cells react to the alphas? And one other part is the cell viability. So how strongly do we kill the cells? Do we kill all the cells? Are there different sensitivities? And when you have the caspase-3 assay, so you check how do you induce the cell death? Is that an apoptosis, or would you rather have a necrosis, or what is the mechanism? And to go a little bit more into this mechanism, so of course we also look at what happens with the alpha radiation in the in vitro. And so there are really some special considerations. When we first started working with alphas, it's you have to get used to that it's not just one decay, but you have so many alphas, so many daughters that come with also more alphas. And that it's not just alpha, but you also have beta and gamma. And the alphas are Yes, and something it's really good that they are so easily shielded, but for experimental setups, it really faces many restrictions. And on the upper right, you see when you just add your alpha into a liquid, then you will probably get all kind of effects, but many of the alphas will be too far off from your cells, so you will not elicit any response. And on the left, you see a schematic. What we've been trying to do is, so we are using this transfer system. We have the alphas coded on the bottom of a well, and then we have a well with cells with a very thin membrane placed on top, and there we kind of have exposure of the cells to the alphas. And we've tried this, and this you see on the right, where we have exposed the cells to 8.2 grays. And you can see when you have an overlay of the double strand breaks and the DAPI that you have this purple spots there showing double strand breaks in the cell nuclei. And when we shield the alpha, so we put a thicker plastic on top of the alpha, then you see that these dots disappear. And that's kind of the same as we have when we don't expose at all. And now, of course, it's the question, and here we had really very valuable help from Balázs, was so how much dose do we then actually deliver to the cells? And he did some really nice Monte Carlo simulations and calculated for us, so what is the dose when we use radium-223, how much dose do we deliver to the cells? Because that gives us also some kind of measures on how the different systems work, on when we use different transfers, when we want to compare. Because also the system is very good because we wanted to compare different cells. And one problem is when you have targeted radiotherapy or you use a tracer, then you have also the issue of having different amounts of antigen on your cell. So it's not only that you have a different cell line that has different genetic background and potentially could show different reactions, but it's very hard to then standardize to how much dose I actually deliver. So this is why this transfer system is very convenient to allow a more general exposure or a more normalizable exposure to the cells. And on the very left up, you see that is what is happening. So it's a comet assay, and you have this big round blob that's just a cell nuclei, and these other two comets, that's really in the back of the cells. That is really DNA fragments. So it's the alphas really break apart the DNA, and then you have many fragments floating around, or not floating around, but you have many fragments of the DNA, and that is what kind of finally makes the cell die or kills the cell. And then so what we have here is we are then trying to understand what does the alpha do to a cell. And for example, we look at the amount of DNA damage. And here we have in the middle, upper middle, we have 4 different cell lines. And we have checked how much damage do we do to the cells. Is there a difference? And that with increasing amount of time. So we have 2 hours, 4, 6, 8, and 24 hours. And then we have a color coding system. So blue is almost no damage, then green is more, and the very red is a lot of damage. And as you can see, of course, the longer you expose, the more damage you have with the cells. But also very interesting, this varies. And for some cells, it's extremely interesting. After 24 hours, you see this increase in blue, meaning that there are actually cells that don't have any DNA damage at all. And so that is really an interesting phenomenon which we're investigating. On the cell cycle arrest is also very interesting. On the right, there are 2 different cell lines, the 22RV1 and the HC116. And what you can see for the 22RV1 is that you have a change in cell cycle between G1, S, and G2, but that is very little, this change. So you see a little bit decrease in G1 and a little bit of increase in G2. S phases, it's not changing much, but its picture is very different when you look at the HTT116. Here you see for the stronger and longer exposures, you see a very strong increase of a G2 arrest and a very strong G1, a decrease in G1, and also S-phase is going down. So these two cells are reacting very different to the same amount of radiation. And we would like to understand why is this the case, and would this help us to understand and tailor more radiotherapeutic therapy. And finally, on the lower right, on the lower left, you see that we are also looking at cell death. And here we found this interesting phenomenon that we did two scenarios. So one is we kept the time constant and we increased the dose. And then we kept the dose constant and increased the time of exposure. And what you can see by just looking at these graphs, it's very different. So we are learning there a lot. And I'm too long. So I'm speeding up a little bit. So we have when we have a very good trying to find a targeting molecule, we also look in vitro, very important. And you see that we're checking high expressing, low expressing, and PDX models. And here we see in red always the therapeutic efficacy. And also one interesting feature for alphas is there is no linearity. So here we have actually doubled the dose and there is no doubling the effect or no halving the effect when you go in the other direction. Interesting. Why is that? And we're also looking at bone metastases models. And here also this alpha therapy is very efficacious as we actually expected from radium-223. And of course, we're also looking at side effects. And we're monitoring the body weight. And then we do also biodistribution experiments. And here's a comparison where you have on the left the targeted compound and on the right an untargeted compound. And you can see that there's a very different profile. So we have on the very left, we have this huge amount delivered to the tumor. And then you see this lower decrease in the blood, and there's also some accumulation in the various organs. Whereas when you have something untargeted, it just very randomly distributes, probably with the blood and where it just likes to stick. And of course, from this, so this is of course influenced by the protein dose. And of course, this will have, as you see here on the right, effect on the white blood cells, for example. So here you can see that when you increase the dose, you see over time, you see a decrease in white blood cells. So you have some hematox and you see a slow recovery. So also something interesting. And what we're also trying to investigate is what is happening in the target tumor tissue and what is happening also in the non-target tumor, in the non-target tissue. Here are two examples for gamma H2X. So really damage or DNA damage or DNA breaks and the caspase induction. But what we've recently acquired, and that's going to, I think, being really, really interesting, it's a gas detector for real-time detection of autoradiography. And you can see that with the different compounds, you have various measures of these compounds. So you have on the right in the tumor, you see differences in the biodistribution in the tumor over time. And very interesting on the right, the kidney localization. So compound 1 is easily distributing quite evenly, and compound 2 is really very much in the cortex of the kidneys and sparing the internals, the mark of the kidneys. So this is something where we hope that we can have kind of a, probably would call, a micro dosimetry to see really where do we deliver the dose and what can we learn from this dose. And here I come with my last slide. And you've heard all this. And if there are any questions, feel free to ask. And I'd like to thank the research team and development team in Oslo and Berlin. And I'm sorry that I was a little bit over time.
Yeah, thank you, Sabine. Very interesting talk. Very nice also to hear more about what's happening inside the cells. I'm not a biologist, but a physicist, so I look to the material quite differently. So I learned a lot. Thank you. Let me see. if there are any questions published. So far, I don't see any questions. Maybe in the meantime, I can give you some feedback about the poll we launched just before or just after my meeting, my lecture. And then you see that if you look into what you believe that we should focusing on, you see that, well, none is answering that we should do none of these data, but there is a mixture between what the role of Eurados should be in the involvement um, for the development of internal dosimetry procedures in radiopharmaceuticals. So very interesting. Thank you very much for answering these questions. So I would like to move on to the next speaker. The next speaker is Sander de Groot from Energy, and he will tell something about the field lab for advancing nuclear medicine. Sander, please go ahead.
Okay, Ramona, thank you very much. So as introduced, I am Sander de Groot, Program Manager of Medical Isotope Development at Energy Pallas. I'd like to thank Eurados for the invitation to present what we are doing typically at Energy in Pallas, and also related to the developments on medical isotope production mainly. And we have the field lab for that specifically, which I will introduce. Basically, what we are doing in pattern mainly is producing medical isotopes, and to a very large extent this is focused on diagnostics, and to a large extent diagnostics is focused on molybdenum-99 and technetium-99. However, we see, and also in our production, that the demand for therapeutics is increasing drastically, and this is also where we see a very large future for isotope production. I think I do not need to go into detail in this. This has been explained by the previous speaker. This is typically the therapeutic applications that have a very large future as we see it now. It's where you combine a basically harmful isotope to via a chelator to a targeting compound, and this targeting compound is able to connect to receptors or to specific cancer cells, and in that way you administer a very local harmful dose with the intent to make that happen only at the tumor site and not anywhere else. And in this way you can very specifically target cancer cells and eliminate them. And if this is done well, then this is a very local type of radiation that will have hardly any side effects. But of course that is where a lot of development is still required. I just gave you an example on lutetium, specifically intended for net tumor applications, but there is a huge amount of different types of compounds are under development, and many of those related to lutetium, which is, as you well know, a beta emitter. And as you can see, one isotope can have many different applications, and each of these targeting molecules has a different type of working, a different type of cancer it targets. And this also allows, or at least provides perspective on the future, where many different targeting compounds can be targeted for specific cancer types and also the specific patients. So this allows for personalized treatment. And as said, if this is done well, so if the targeting is very specific and very efficient, this could be done with limited side effects. Because of this perspective, we expect a very large growth, and we also already see that happening. And this could be based on lutetium as maybe the workhorse for future therapy, although there are many other isotopes that could take the place of lutetium or be complementary to it. For example, other betas that have a specific half-life or energy. And of course also alpha emitters are of large interest. And in my presentation, I will also highlight a bit the potential perspective of Auger emitting isotopes. So Energy Pallas is one of the largest producer, if not the largest producer of isotopes worldwide. It's not that we are not providing any end products. It's we have irradiation capacity and produce almost 30% of the worldwide demand. This is to a large extent molybdenum, but we have a similar type of market for the therapeutic isotopes, lutetium, iridium, etc. And based on that position, we also want to prepare for the future, specifically on the therapeutic side. And that means that on the irradiation side, we are aiming to replace our high flux reactor with a new reactor called the Pallas reactor. Also our processing capabilities, which are now basically the molybdenum processing facility, would like to extend to therapeutic processing as well, so that we can take the products from our reactor and separate and purify them and turn them into a good semi-product or API. And we also do innovation, because innovation is needed both on the production of these isotopes, but also on helping out on clinical trials. And with all the nuclear infrastructure available in Patton, we can play an important role on the innovation side as well. And for that, we have established the field lab. So if you look at the whole supply chain, there are many locations in the supply chain where dosimetry plays a role in different capacities. And just to guide you through this, we initiate the production of isotopes generally with a target material. And this can be an enriched isotope, for example, and some of those are really expensive or very scarce or both. And then you activate them, so you add protons or neutrons to it to turn them into radioactive material. And after irradiation, you separate and purify these to come up with the pure isotope in solution, which we generally call a radiochemical. And this is an active ingredient that can be used for product manufacturing. In this separation purification process, because these target materials are so scarce or expensive, there is in many cases also a recycling route envisaged. At pharma, the semi-product is combined with, for example, a targeting molecule, and then you have a medicine or a generator. That can also be the case for some of the isotopes. We can foresee that the half-life is short, but mother isotopes provide a longer use of this isotope by turning it into a generator. And this is then transported to the hospital, where it can be administered to the patient. In general, just because this is for Eurados, I just wanted to highlight a number of aspects that from a dosimetry point of view are quite important. Of course, there is occupational exposure in the whole production route. We're talking about infrastructure and not new infrastructure also to be established, and the protection of the employees and operators is very important. Some of these isotopes, when they're coming fresh from the reactor, generate a very large high-level irradiation field. After purification, it can be limited, but you need to take care in your processing facility to be able to handle this, and dosimetry plays an important role there. Of course, in the whole chain, there are different locations where nuclear waste or radioactive waste is generated. This is, of course, in the processing facility, but also in the production facility and also in the hospital. And of course, also the patient can generate radioactive waste streams that need to be cared about and need to be mitigated or managed in a proper way. So this environmental exposure needs to be addressed. Then, of course, the patient, him or herself, medical exposure. So what is the balance within the patient between the toxicity that the compound is generating and the efficiency? And this balance, of course, should be to the benefit of the patient. And then, of course, there's also occupational exposure at the hospital site. So the impact of new nuclear medicine treatment in the hospital. Hospitals are just beginning to start with adopting nuclear medicine treatment. So there's a lot of management and infrastructure needed to be able to manage that within the hospital. And also the employees in the hospitals need to be protected in a proper way in case the amount of treatments with therapeutic isotopes increases. So within this supply chain, we have added a specific part on the innovation side, which is the field lab. This is a collaboration with academic partners and industry, where we look at new isotopes, make them available within the field lab, but also we are currently establishing within patent infrastructure that we can also do labeling and ultimately also at the GMP level, so that we can also support clinical trials with end products. But of course, all small scale and intended to support the development. And some part of the development is also really on the processing side. So how can we turn something that has been irradiated and is a mixture of all kinds of stuff that is generated in the reactor, for example, how can we take out the isotope and get it to the right purity? And also some isotopes require specific development on the irradiation side. So how to safely irradiate it and introduce it into the reactor and take it out again. All these aspects are covered within the field lab collaboration. So we aim to enhance, accelerate development, and also make these new isotopes available in a nice radiochemical, so a pure and radiochemical of the right form. But also we try to make facilities available for also producing radiopharmaceuticals. It is very important, and this I think is where ORADOS really has an important role to play, that these occupational and environmental and medical exposure risks need to be assessed as early on as possible, including mitigation strategies, to be able to not have a large risk blocking the implementation of these new treatments in the future. So for effective and successful development, an early on assessment on dosimetry, on all the aspects I just mentioned, I think is very critical. So I now have a number of examples that I would like to share with you, and with some recommendations from our side for dosimetry to focus on. Well, this was already mentioned also early on. Actinium-225 is an alpha emitter with a lot of perspective. It's regarded by many developers of new products as a key isotope with a very large perspective. So these type of images are generally also used to demonstrate its efficacy. Nevertheless, these pictures also show some other side effects that are less favorable. I highlighted this with these red circles, where you can see that the salivary glands of this patient really have suffered from the treatment quite significantly. So there is side effects that need to be taken care of. And also on actinium-225, even though it is regarded something that can be implemented very quickly, a significant amount of research and development is still needed, at least in our view, specifically on the targeting efficiency and also compound-related off-target effects. So to couple dosimetry with compound biodistribution and kinetics, and especially the off-target effects, could be key for the future implementation and justification of these type of treatments. Then there are, of course, more alpha isotopes. Thorium-227 was mentioned in the previous speech, which is, of course, also a high potential alpha isotope. I made a list here of many of those, and how to select the right one. Yeah, it's basically a balance between decay time and the damage profile. Of course, also how the isotope is bound to the targeting compound, and also how this is bound on the longer run. For example, after one decay, generally the isotope is no longer bound to the targeting compound. So how will the daughters of this isotope distribute, and what type of damage will could it give in off-target areas within the patient? And yeah, this balance between the right isotope and the behavior of the compound is very critical. And one isotope might be very suitable for a certain compound, for example, a longer half-life, and that sticks to the targeting area for a very long time. That is fine, but some of the targeting compounds act quite quickly and also leave the receptors quite quickly, and for that a longer-lived half-life isotope is maybe less suitable. Then, of course, if you move to a short-lived isotope, then you have the challenge to produce, process, distribute, and get this into the patient. with the right specific activity. And also availability and costs of these isotopes are quite critical. Generally in an R&D stage, small amounts of material can always be retrieved somewhere. But if you do not think about the longer run and that can this be upscaled, can this really be produced in large quantities in a proper way, is also something that should be taken into account in the R&D phase. Because many patients will get treatment. We have nice pictures like the slide I showed before. There are a lot of expectations raised, but if you cannot really supply, then you're basically raising expectations that cannot be met. But with regards to these recoil effects, it's I think also very important from a dosimetry point of view to take it into account. So targeting efficiency, but also decay-related off-target effects are quite important. So what are these free isotopes due to recoil actually doing? And therefore, dosimetry coupled to biodistribution and kinetics of decay daughters is also an important aspect of future alpha therapy. So one of the isotopes that we are looking at quite seriously, also because our field lab partners have indicated that this could be a really nice isotope to look at, is lead-212. It is basically a beta emitter that subsequently sends out an alpha. Beta emission can also lead to recoil effects, but we have clear indications that there are chelators that can withstand the recoil of beta decay quite effectively. That means that the alpha is still contained within the compound. It has a half-hour half-life, so this is really short, and that is suitable for certain types of targeting, definitely not all of those. And also there are concerns that this high activity that needs to be introduced into the patient also could lead to kidney damage, for example. And maybe that needs to be protected in these type of treatments because you initially bring a lot of alphas in the patient. Of course, it also disappears quite quickly, but this initial hit should be managed in a proper way. It has longer-lived mother isotopes, and this enables centralized production, which is convenient, and it can be distributed via generators. So basically, at the hospital site, you can extract the lead-212, combine it with your targeting compound, and turn it into a medicine. And also, there are many ways to produce these mother isotopes. Ultimately, the mother isotope is stored in 228 with a half-life of almost 2 years, so can be produced centrally quite conveniently within different ways. So this is all quite nice, but there is also a pretty large downside to lead-212, which is that in the thallium-208, one of the daughters in the decay chain of lead-212, has a very hard gamma, higher than 2 megaelectron volt. It's something difficult to shield. And for the patients, most likely not that much of an issue. Damage, I mean, the dose because of that gamma will not be very significant, but especially environment needs to be managed. So if many patients get these treatment and they stay in the hospital for a significant amount of time, all these, the population of patients in such a hospital can lead to a very high environmental dose, and workers need to be protected from that. And this is a challenge for this particular isotope. Then another types of isotope that could be really relevant are Auger emitters. And on the right, you can see an image where very basic the damage profile of the different isotopes is depicted. As you can see, a beta creates damage here and there over quite a significant range. And alpha emitters have a much more dense damage profile. And this is also where you can see that the beta emitter could lead to single-strand breaks that basically could be repaired. And this is also why beta emitting therapies show resistance. Cells can repair themselves. So beta emitting therapies ultimately get less effective because of that. And alpha emitters lead to double-strand breaks quite easily, which is extremely effective. But Auger emitters, despite a very small amount of energy it deposits, can lead to a significant amount of DNA damage if it is well targeted. And this, for example, provides perspective of terbium-161, which is similar to lutetium, but also has Auger emits Auger electrons. And this, if located well enough, could lead to a lot of damage and could be very, very effective. Of course, the challenge is how to get that Auger emitter in the right location. And this is where very specific dosimetry could come into play. where you really would need in-cell nanodosimetry in combination with in-cell biokinetics to be able to predict or at least do recommendations on how Auger emitters should be used in therapy. And also off-target effects need to be assessed, because this Auger effect could also, of course, be happening in cells in organs if the Auger emitter is able to reach DNA or cell membrane locations. Am I still on time or am I over time? I cannot see it.
You're already a bit over time.
Okay. Well, this is my last slide. So, and basically it's a summary of what I already said. And maybe for the future, just an overview that molybdenum on diagnostic side is already a workhorse for diagnostics for decades and will be for decades to come. And pain relief isotopes are already well known also for decades. The next big thing, but it's already happening, is lutetium. And terbium, with its Auger emitting capabilities, could be an extension or maybe even a replacement of lutetium in the future. Then alpha emitters are, of course, very high perspective, but need to be assessed in all these aspects that I mentioned on dosimetry. And maybe Auger emitters, if well targeted, could be the future as well. And yeah, all of these developments bring a large amount of opportunities for dosimetry and, of course, challenges, which I summarize here, but they're basically already mentioned in my presentation. So I will not go into detail because I'm over time. So thank you very much. That was my.
Okay, thank you very much, Sander. Very interesting to hear more about the radiochemistry and challenges you think we have to solve a bit within our task group or other task groups. I see there are some interesting questions inside the Q&A box. Maybe you can try to answer them by typing, because we are over time. I would like to go to the next speaker. The next speaker is Maarten Ooms from SCK Sen, and he's going to tell something more about the role of terbium 161. Maarten, please
Yeah.
go ahead.
Okay. Hi. So yeah, my name is Maarten Ooms. I'm from SCK, a radiopharmacist from training. So yeah, don't expect hardcore dosimetry from me. But so what I would like to I would like to use the next 15 minutes to draw your attention towards terbium 161. Which the past few years is emerging as a high potential alternative for lutetium in targeted radionuclide therapy. So if you look at terbium itself, terbium is actually quite nice element to use in nuclear medicine, as it contains several isotopes with nuclear medicine applications. So you have the terbium 152 and the 155, which have imaging properties. So you have a beta plus emitter and you have a gamma emitter, so you can do PET and SPECT imaging. However, on the other hand, you also have the 149 and the 161, which can be used as therapeutical radionuclide, so alpha therapy and beta therapy. Additionally, it's not that much nuclear medicine, but terbium itself, natural terbium, also has some fluorescent properties, so you could even use fluorescent luminescence or that could also be an application of terbium itself, non-nuclear then. So yeah, the advantage. This is some data from the group of PSI who has been working on terbium for quite a while now. So yeah, the advantage of this summarize a bit the advantage of using terbium. So you can have terbium 152, you have terbium 155, terbium 161. They're all chemically the same. So chemically speaking, there is no difference between the different compounds, yet you can have different physical properties. So you can use different decay results. So that's a bit the major plus side about this terbium, that you have also brother and sister isotopes that you can use for imaging using the same chemical compounds. So, and that's a bit of an advantage compared to some other isotopes where you have different isotopes to do the dosimetry, for example, than or different elements to do the dosimetry than what you do for therapy. So that could be a major advantage of terbium. Now, a bit very short on the production of terbium-161. So terbium-161 is produced starting from gadolinium-161. So we basically seal these ampules, this powder in ampules, which we put in our nuclear reactor to get a nuclear reaction. That yields terbium-161. So then we get irradiation ampules back, irradiated ampules. We need to crush them and then apply radiochemical separation to extract the low quantities, chemically speaking, then of terbium from a large excess of gadolinium. So the terbium we can then use for radio labeling, while the gadolinium we're working on having some recycling process to reuse it, since it's a quite expensive and a quite valuable asset. We don't want to just discard it, and we want to try to reuse it again. So that's in a nutshell the production. Now, why focusing on terbium-161? I think it's been mentioned here today before earlier as well. So it is an alternative for lutetium-177. So chemically speaking, they're both lanthanides, so they behave quite similarly. You can use a lot of the same chelators to conjugate it to radiopharmaceuticals. Also, a lot of the physical properties are the same. Yeah, they both have a half-life of approximately 7 days, give or take, yeah, a few hours. They have beta emitting. They do beta emission with similar energies. However, the major difference between the two is that terbium emits a lot more Auger electrons, and these low energy electrons are a very powerful tool. So they dispose a lot of those on a very short range, so which indicates terbium could have a much higher potential compared to lutetium-177. Now, if you look at, yeah, as I mentioned before already, PSI has been working a lot on this before. So, and as an example, what they did is they made some folate derivative. So, if you want to compare both, they made a head-to-head comparison, the radio labeling the same folate derivative with lutetium and terbium-161. And then the advantage is that you can, yeah, directly compare them. So if you look at the pharmacokinetics, here at the right, you see an image which is basically a time activity curve of your radioactivity over time in the blood of a tumor mouse. You can see actually that the pharmacokinetic profile is quite similar. So you don't really see major differences in pharmacokinetics, which is also what you expect between the two, since, yeah, they're both lanthanides and the chemistry is similar. However, if you look at the therapeutics, so they also looked at some therapeutic parameters between the two. So the top figure here is a cell viability it's assay, where they could clearly see that that the effect of terbium actually occurs at a much lower dose compared to lutetium. So this indicates indeed the high potential of terbium-161. And also in the survival curves, they did some tumor survival model where they treated tumor models with terbium and lutetium compound. You can clearly see the difference between the two isotopes. So that's really the effect. Chemically, the carrier is exactly the same. It's only a difference in the isotope. So that gives an indication that terbium really has a high potential. Also on a very popular target, the PSMA, which is, yeah, booming in nuclear medicine these days. They also made a terbium derivative that really shows high potential and is a promising candidate. So you really see an increased survival with terbium PSMA compound. Now, of course, this was a bit, yeah, the state of the art and how it started. The problem is that, yeah, terbium-161 so far, the research has always been focusing on well-known carriers like the folate or dotatate or the PSMA compounds, which are quite heat resistant. So all the radio labeling methods that have been used high temperatures, 90 degrees, 15 minutes. However, within nuclear medicine, there's a big shift towards, yeah, also more, yeah, innovative carriers, some, yeah, or at least more biologically unstable carriers like antibodies, nanobodies, or any antibody fragment. So the problem with these is that they're actually not that yeah, not that they can't stand these temperatures that well. So you cannot perform radio labelings at 90 degrees if you're using a nanobody. So and yeah, then that led here at SEK to start a quest for better or lower temperature radio labeling methods. And in an attempt to do that, I'll quickly move my to show Erwin. So in an attempt to do this, we actually started up a PhD project together with the Radiopharmaceutical Research Lab of KU Leuven. And yeah, for that, yeah, Erwin Kessel's a PhD student. He synthesized a few compounds and tested a few compounds for their radiolabeling properties. Um, with terbium. So and there we focused really on low temperature. So we had to make sure that the temperature was compatible with heat sensitive biomolecules. So if you look at this figure, yeah, you can see the DOTA chelator, which is often used or was often used, works at these lower temperatures, but you don't really get quantitative yields. Um, However, some other chelators seem to be a lot more promising chelating the terbium, which is important. So we looked at how to get it in. But of course, what's at least as important is also looking at how fast does the terbium get out again. So we need to make sure that the stability in vitro and in vivo is decent, so that we don't lose the terbium the moment we start injecting. So if we look at the same chelators here, so we did an in vitro stability in both human serum and in PBS. So the PBS there, it still seemed to be quite stable. However, if you look at the human serum DTPA, that's even after 24 hours, you start already seeing a significant decrease in yeah, association of terbium with the chelator. So yeah, the reason probably is because DTPA is known to be a very good compound to complex everything, which also indicate or means that the moment you bring other metals near it, it will probably have a lot of competition between terbium and any other metals in the human serum. So this is an in vitro setting. Next, we thought about trying to find a way for an in vivo proof of stability, and therefore we conjugated all of these chelators to human serum albumin. Um, the major advantage of this albumin is that it's a protein that does not really migrate to any tissue, so it stays in the blood for circulating a lot, and that also is perfect to see where you see accumulation. That means that probably if you see accumulation at some places, it means that you probably have loss of terbium from your chelator. For example, the DTPA compound, which I'm already mentioned as not being very stable, what you clearly see here is that in the bone you see gradual accumulation over time of radioactivity. So this is really important to look at. Certainly, if you know that, we also did this biodistribution studies with free terbium, where you can clearly see that the terbium itself goes to the bone. So basically, that accumulation of terbium we saw with the DTPA means that the stability of the compound is not really well. So that's basically, we're now trying to apply some of these chelators to innovative carriers. I'm not going to go into detail about that because time would be a bit too short. However, what we looked at having a good chelator. That's very important, but also what might be even more important for from a dosimetry point of view is we need to maximize the Auger potential, because the potential of terbium over lutetium is only valid if you can really use these Auger electrons. So and therefore we need to look, I doubt I have to explain this to you, but indeed the beta particles, they travel or range of several millimeters. So they basically, one beta particle can hit a lot of cells. However, for the Auger emitters, since they have such a short range, it is actually crucial to have them really, they don't even, you need to have them in the cell very near to where they do the damage to actually have the potential. So and therefore it's important that internalization and intracellular targeting is crucial. So one of the tests we are doing, which is a um, a project we started off with. Actually was initiated by the University of Lisbon, by the group of Antonio Paulo. So they were looking at different ways to try to target radioactivity to the cells, and this would be one of the perfect examples where terbium could be beneficial. So therefore we started to collaborate them, supplying them with terbium so they could try to look at specific delivery of these radionuclides into radiosensitive organelles like the mitochondria or the cell nucleus. And therefore, in doing that, we try to enhance the radiotherapeutic effects at lower dose and maximizing the potential of Auger electrons. Now, so as a conclusions, terbium-161, is a high potential alternative for lutetium-177. So with the Auger emitters comes a great power. However, you need to be able to target them well. One thing is, so far terbium, a lot of work has been done um, on the therapeutic level to look at the therapeutic efficiency. However, the dosimetry and the real detailed dosimetry still needs to start. For example, at SEK, we are currently starting 2 new projects where they really want to do this dosimetry on terbium 161 and really have good models for the 161, including the Augers. So what is still a problem though is that terbium, it's not really that available these days. So there are not that many groups working with it, and that creates a bit of a vicious circle. So due to limited supply, there's also limited research opportunities or groups that are able to work with terbium 161, and that results in, of course, that then it brings it to clinic slowly, which in return again decreases the interest of suppliers to start working on it. So, and that's why SEK and I assume also other people are trying actually to break that circle. So we've been producing terbium and collaborating with several partners to try to get terbium out of that niche market and try to really get it into mainstream. So we will continue working together and working in-house on trying to get terbium to the market and to more application. So finally, I think I would like to thank you for your attention. And if there are any questions, I will gladly answer them or in the chat or later on in this presentation.
Thank you, Martin. Very nice presentation. Good to learn more about terbium and how the chelator research is conducted. Currently, I don't see directly any questions for your presentation. Maybe at a few moments later, because I see there is a bit delay in questions coming in. So for the meantime, I would like to thank you for your presentation. Very nice. And I would like to move on to the next speaker. The next speaker is Thierry Stora from CERN, and he will tell something about the PRISPA program he is working on. Thierry, please go ahead.
Yeah, hello. So actually, some very nice to be invited to this very interesting session on therapeutic radiopharmaceutical and dosimetry. So my presentation is recorded, so I am there, but you'll see my little virtual myself during the presentation. I'm happy then to answer questions in the chat or whenever.
Okay, thank you.
Hello, everyone. This is my pleasure to introduce you today with the PRISMAP project. I'm Thierry Stora. I'm a senior physicist at CERN, the European Organization for Nuclear Research based in Geneva. I'm the coordinator of this new European infrastructure program from the European Commission. And I will share with you today the details on this starting program, which is aimed at providing novel radionuclides for biomedical researchers. How will PRISMAP contribute to the field of novel radionuclides for research in medicine. So I think we have to start to look at the statement that is there a way to progress on the difficulties that is found to provide radionuclides and non-conventional radionuclides for nuclear medicine research, and for most of those they can be available and produced from large European infrastructures. So, of course, we are not speaking about technetium or fluorinating for ADG, not even gallium or 177 lutetium, but rather what is in the pipeline for developing new radiopharmaceutical. I mean, this is certainly not a new idea. We all know the story that when Madame Curie found radium, she very rapidly identified the potential of this actually alpha emitter to provide treatment compounds. But as well, we all know that to get a radionuclide into even a clinical testing phase, this is a long way. And so the rationale of PrisMaP is indeed to provide a tool to federate a consortium of large-scale infrastructures in Europe and to promote the interactions and development so that there is a common service which can be provided to medical researchers in Europe. So the ambition of PrisMaP is to therefore provide access to these new radionuclides and with new purity grades to fragmented researchers community. They can be developing radiopharmaceutical, new imaging technologies, new biological targets, and the way will be through a single web interface, prismap.eu, to be able to submit a project and to get information from this interface. So we are aiming at enhancing the clarity as well and the regulatory procedures for the shipment, for the manipulation and for the disposal of these radionuclides, and through as well generating some new data, nuclear data, eventually biological data made available so that the radionuclides are better used. On the long term, we'd like to find a sustainable structure so that PrisMaP, which is initially in the initial phase of 4 years, can last actually on the long term. It's certainly known to a vast majority of the scientists, researchers, medical doctors active in radiopharmaceutical development or technology development, that indeed to produce and find a new radiobioconjugate, new radiopharmaceutical, either useful in diagnostics and of course now more and more into treatment, you need to have the proper radionuclides. And if you just take the field of targeted alpha therapy, we have actually a challenge to supply those radionuclides at a sufficient scale. And if we take only the 225 actinium, but actually there is a number of other alpha emitters which have interest. And I can speak, for instance, of terbium-149, which is a radionuclide with alpha emitting properties. There is needs to have increased supply in Europe and beyond. So, of course, these radionuclides, they are classically supplied by, let's say, nuclear reactors, research reactors, or small cyclotrons. And then in Europe, we do have a few larger cyclotrons, accelerators, hosted at the fundamental research centers. And then PRISMAP beyond, creating starting a consortium of research reactors, high power, high energy cyclotron, is as well integrating in the consortium mass separation facilities, which by this physical mass separation technique has the capacity to change, for instance, the purity grade, the specific activity of some radionuclides, for instance, those sometimes available from nuclear reactors. So the consortium of PRISMAP is indeed composed then of the major nuclear research reactors in Europe, so that Institut Laue-Langevin in Grenoble, or the BR2 in SCKCN in Belgium. So complementing the nuclear reactors, we have high power, high energy cyclotrons, such as Aronax in France, or Injector 2 at the Polscha Institute in Switzerland. As I'm coordinating MEDICIS, PRISMAP is put a certain emphasis on the capacity of having a mass separation facility, so that we can exploit high energy proton beams, such as that available at CERN with CERN MEDICIS. And we'll have actually future facilities coming online, such as Isolfarm at INFN in Italy, which will be able to extend the mass separation facility capability. And a longer term, we may have, for instance, Isolamira in Belgium. This consortium of major nuclear centers, accelerator centers, and mass separation facilities is then complemented with a network of biomedical research facilities, hospitals, which can do preclinical and clinical testing, such as the nuclear medicine department in Munich, or the hospital in Lausanne, or EVZ laboratory, for instance, in Denmark. And so they both have the license, the proximity to exploit some of these exotic radionuclides. So we may ask ourselves of what is then the scope, the need of these non-conventional radionuclides. And then if we look simply into a range of treatment radionuclides with hours to days physical half-lives, we see that with their emitting probability of Auger electron alpha or beta particle, the range of emission can go from subcellular to let's say a monocellular, to a small lesion, to large lesions, and then the hierarchy is not necessarily as well scaling with the established and not established radionuclides, where we could, for instance, find Lutetium-177, which is in between, because it's no more an emerging isotope, but definitely, if you think that some grades can be produced so that you are not co-producing a long-lived impurity, which is posing challenges in the waste handling and the patient management. Then we actually already identify that you want to precisely know the half-life and the emission property, probably very precisely for the Auger emitters. So this potentially interesting new treatment isotope can be used, of course, in theranostics and combining with imaging properties. One has to understand how then the biomedical centers, which can host the researchers which can benefit from PRISMAP services, what are somehow the services and the capacity which is offered then and will be offered through a PRISMAP granted project. And so basically once the researchers have identified their molecular target, then there's a range of techniques which need to be applied with these novel radionuclides, which is from labeling to biodistribution, preclinical studies, and have regulatory documentation done so that you eventually reach pilot clinical phase. And all that is actually made possible in some of the Institute of PRISMAP that complements this network of unique facilities in terms of research reactor or accelerators. So here it's a list of so-called day one radionuclides, which will cover combination of imaging and therapeutic radionuclides, radiometals, either copper 64, 67, 44 scandium, 47 scandium, but goes into radiolantanides and ultimately to alpha emitters such as 211-statin. So this, you see, indeed covers a wide range of modalities in nuclear medicine, and present level of offer of these radionuclides can span from purely research preclinical stage for some of those radionuclides up to some more mature isotopes, so that you can have good laboratory practice grade for 225 actinium from the Joint Research Center in Karlsruhe, or up to GMP grades if you get 64 copper from Aronax or DTU. Of course, this is very dependent of the type of research project which can be received and streamlined in the consortium, but definitely the range of the day one radionuclides is already covering the different modalities and with a range of half-lives and chemical properties. So that project is expected to be received in either developing protocols, imaging techniques, checking impact of radionuclide grades on the outcome of the research project, or simply bringing these innovative isotopes in a, I would say, a classical theranostics approach. And as I mentioned, we are mostly dealing with studies on, or will be receiving project for studies on cells. Preclinical is the major, I would say, expected research project on this platform, on this PRISMAP program. But we have the capacity to host a few pilot clinical trials when the grade and the project is ready. To further highlight the type of project which might be received in PrisMap, and there is, for instance, a topical issue which we co-edited with Professor De Christopher and Professor Pryor from Innsbruck and Lausanne, where you see that details can be reported on, for instance, how the mass separation facility Emitters has been operating for the past few years, and how this is, for instance, enabling either through the network or through the isotope production from Emitters itself, some interesting developments, either providing non-carrier either high specific activity erbium-169, which is otherwise used in a dilute form simply for pain relief in joint arthritis, and where the possibility to make a physical mass separation can lead to preclinical investigations on radiobioconjugate. And we have a similar trend with developing terbium radiobioconjugates, because terbium can really go from let's say an equivalent of lutetium-177 with a beta treatment isotopes, but then going to 155, 152 terbium to SPECT imaging or PET imaging, and up to ultimately 149 terbium for targeted alpha therapy. And in that respect, in this topical journal, we had a very interesting, let's say, a short paper, but very well addressing the non-simple aspect of radiobiological effect on using targeted alpha therapeutical agents. And as a final example, this is of course addressing what is the regulatory framework and the possibility to use these novel radio isotopes and getting homologation as investigational medicinal product. And here this is just a further example showing how 152 terbium as a PET emitting radionuclide, and there will be other presentations in this session on these different aspects that I've just mentioned. How they it can be used with a antibody-based radiobioconjugate, where the physical half-life of the PET emitting 152 terbium is much more adapted to a biological half-life of the bioconjugate to get proper enhanced imaging and biodistribution. And then in the idea of theranostic, you would then be able to investigate the radiobioconjugate with an alpha or beta emitting terbium isotope. So PrisMap will soon launch a call for projects. We'll have twice a year a call for projects with published selection criteria, but basically this is excellent science which will be the main criteria, and we expect groups from academia, from university, hospital, from nuclear centers to be able to submit their project, even from SMEs, if this is found to fit the criteria. And the services will be provided then free of charge, besides just the shipping of the radionuclides. So this is how the project will be streamlined, and so we expect to then be able to address the new treatments nuclear medicine in Europe. With that, I thank you very much.
Thank you, Terry, for this very interesting talk. I don't know whether there were especially questions addressed to you, or that you would like to have any additional remark to your talk?
No, actually, there was some which were asked to Martins, which he answered, and which I answered, which were quite relevant also to this talk. So,
Okay.
if there is any further, do not hesitate, of course, to put that in.
Yeah. Maybe some other questions will come in later. But it was very nice to hear something more about the Prisma project and about it for the upcoming goals for delivery on the several radionuclides. Was also here to hear more about the non-conventional isotopes that you are looking at.
Right. If you want to do symmetric studies on these isotopes, I mean, definitely you may want to get those from Prisma.
We will keep it in mind. Thank you. If there are no any further comments, I saw in the Q&A sessions that there were a lot of questions coming in, but I think most of them are already answered. So if you would like to, if the other speakers can come online as well, we can see if there are any further discussions we need to have for the questions inside the Q&A box. So thank you all for your very nice presentations. It was very useful to learn more about the work that you are doing on producing and performing research on the novel isotopes. I saw a lot of questions coming inside the Q&A box, and I already saw you have answered most of them. Do you think there are any questions that you would like to comment on or to give additional feedback on? Nope. If not, then I would like to thank you all for your time, for being patient, for being here. It was really nice to have you here. And yeah, I also would like to thank the audience for being present at this webinar. We have prepared 2 more poll questions for you to be answered, and I hope that you would like to answer them. And thank you for your participation in this meeting.